Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
I am welcoming Mike McAlpine to talk to us all. Mike comes to us from Princeton, where he is faculty in mechanical engineering, although you will variously hear himself described as chemical, electrical, mechanical, materials. But rather than reiterate what's in the talk announcement, in which you can read his bio, I'll tell you facts that aren't in the talk announcement. Namely, the fact that Mike and I have actually known each other since we were like 7 years old. And Mike effectively taught me to play guitar. We played in a, I would say, not fantastic band together in high school. We had our moments. And so I can say without much hesitation that Mike is not only responsible for my thriving musical career, but Songsmith would not have happened without Mike McAlpine. I can say that with confidence. So I'll leave you with that. Don't judge him.
That was going to be my intro. You stole my intro. Okay. But I think it's kind of interesting that, first of all, thanks a lot for having me here. And the thing I think kind of interesting, though, is that we keep, our paths keep converging, because it sounds like a lot of you guys are working on starting to interface devices with the body and things like that. And that's actually what we've been working on, too. And it's actually come out naturally from the stuff we've been doing, because a lot of the materials work these days, actually, there's a lot of interesting areas in materials that have to do with interfacing materials with the body. And I'll tell you what the reason for that is, but I think this can be more of like a conversation. So I have enough slides here to cover the full hour, but if you guys have questions, then I'll probably skip some things depending on what you guys want to do. If you want to hammer me with questions, I'm happy to do that. If you feel like just listening, I'm fine with that too. But mostly what I want to kind of impress on you is the fact that you guys are kind of approaching this area from the top, and actually a lot of materials research these days is approaching this from the bottom, because of the interesting dynamics in terms of mechanics and electronics that have to do with materials as you start to interface them with the body. So these are the kind of things that I've been interested in. There is you can imagine a lot of different areas for this. You guys are working on some really interesting ones. And I think the stuff that we're doing interfaces best with the stuff that you guys are doing, which has to do first with sensing. We're more on kind of like the chemical and biological sensing end of things, and interfacing sensors with the body. And then also with mechanical energy harvesting. And so this is something that we've been working on too. And so I'm going to talk about these two. These are the kind of two areas in my group that we've been working on. And so these are the things we're going to be focused on here. But again, a lot of the concepts here are actually pretty generic to any different material. It could be electronic material, it could be silicon. We're not doing a whole lot of silicon stuff, but there are some people doing that. And so if you have any questions about the processing or the materials itself or anything, feel free to just jump out at me and ask. Okay. And I think the general theme here has to do with the fact that if you look at something like a silicon wafer, I assume you guys have seen a silicon wafer before. I'm not sure what level, what kind of, how far.
I've seen a silicon wafer.
Okay. So basically, it looks like this. So it kind of is like a hard glass-like substrate, and so it's hard, it's brittle, and it needs to be processed at high temperatures in order to form single crystal silicon, which is basically what you need to get the kind of 2 gigahertz performance that you see in processors these days. And so we're talking about 1,000 degrees C in order to process this silicon into single crystal form. You can deposit amorphous silicon at lower temperatures that are compatible with, let's say, the body or with plastics, but then you lose a lot of the functionality. You won't be able to get to the high frequencies that you guys are used to, the 2 gigahertz. So when you think about interfacing a material like this, like silicon with the lung, first of all, you have this temperature, we call it a dichotomy. You have the temperature dichotomy, the fact that tissues can only go up to 30, 40 C or a little bit higher than that, whereas this is being processed at 1,000 degrees C. But then you also have the mechanics of it too, which is the fact that tissue is not just flexible, but it's also stretchable too, whereas this is hard and brittle. It can't be bent, you can't bend it very much, you certainly can't stretch it at all, and so you have a dichotomy here in terms of the mechanics too. And so what we're trying to do is basically use nanoscale engineering in order to overcome this dichotomy and be able to interface materials that have high performance, not low performance like amorphous silicon, but actually high performance materials with the body, and that means tailoring the mechanics, overcoming this temperature issue too in order to be able to do this. And so that's that's really the general, this is why it's interesting from a materials standpoint, why a lot of people have been interested in this. First, it started with interfacing silicon with biological materials and then with plastics, and now it's really with tissue and things like that. So that's what we're trying to accomplish here. Okay, so first part has to do with this, what I showed on the first slide. We've been trying to put sensors directly on the body. This happens to be a tooth here. And the motivation for this had to do with this story that I read about Michelle Bryant, who basically, she suffered an asthma attack in a grocery store recently, and she couldn't speak, obviously, because she had this asthma attack. But fortunately, she had this tattoo on her wrist that said that she had asthma. So the first responders were able to treat her because they knew exactly what was wrong with her. And actually, I have a similar situation because I'm allergic to penicillin. And so my grandfather actually wore a necklace that said he was allergic. The necklace didn't fit me, but I have no way of telling people that I'm allergic to penicillin if I can't talk. So people have been getting these kind of medical tattoos in order to inform first responders. And then I saw this other interesting story at the same time, which had to do with this people, for this purely cosmetic reasons, people getting tattoos on their teeth. And this is William and Kate here on the teeth. And so they're getting these tattoos. I have no idea why you would want those. But these can be permanent or temporary. So this interested me because teeth basically come in contact with two biological media, the first being saliva and the second being breath. And so both of those can contain indicators of whether one, certain disease states in saliva or breath can be certain indicators of disease. So we thought about going beyond this kind of passive tattoos, which just contains passive information, to something that's more active, that actually can not only give you information, but maybe continuously monitor your health status and provide some sort of feedback for you. And so if you look at the properties that one would want to accomplish in such a sensor, the first one is that you need to have compatibility. So this means two things. It means that whatever you put on the body has to be compatible with the body, meaning that your body's not going to reject it or form some sort of irritation to it. But then also this gets back to this concept about the materials from before, which is you don't want the material to degrade. You want to conserve the high performance of the material, whether it's silicon or whatever material it is. Conserve it when you transfer it from its native form onto the body. Second thing is robustness. You want it to be able to be interfaced with the body, not easily fall off in some way, and also basically be robust in the sense that the device itself can handle different conditions if you go into different environments, different temperatures, whatever kind of conditions you want the device to be robust there too. Also performance. When we talk about sensors, basically performance, the two things we're interested in. The first one is sensitivity, which means being able to detect at very low levels. And the second one is selectivity, which means being able to detect what you want to detect and nothing else. And then the last thing is, of course, power and readout, which I think is most in the kind of electrical engineering area that you guys are in, which basically means you don't want to have wires, let's say, coming out of your tooth. You don't want a battery on your tooth. You want to have remote power and remote readout too. So I'll give you the punchline here, which is the strategy we came up with recently, which is basically to interface some sort of device here, put it on some sort of temporary tattoo platform that can be dissolved, let's say on a tooth or on your skin or something like that, and then basically tailor it in such a way that you can pick out what exactly you're interested in and reject everything else. And this happens to be a graphene sheet here that we're using as the sensor platform because that's what's providing the sensitivity that I spoke about before. Okay, so let's break this down part by part. And again, if you have any questions about the materials, feel free to ask. So basically what graphene is, it's kind of a hot material these days. If anyone ever uses pencils anymore, I'm not sure. But basically the pencil lead is not lead. Of course, it's actually made of, it's graphite, which if you take in basic chemistry, you know that graphite is basically alternating layers here, basically stacked layers of carbon, these kind of hexagonal carbon sheets here, which are stacked. And so they can basically glide one sheet to the next across and basically glide across from each other. And you can slough off the sheets there. But the thing is, so it sounds like a kind of weak material, but in the plane, actually, it's actually extremely strong material. It has a Young's modulus of about a terapascal, which is almost the same as diamond in terms of in this individual plane here. So in 2004, and also in terms of electronics, there's nothing particularly special about graphite in terms of electronics. But the individual sheet, this graphene, which is the individual sheet, has amazing electronic properties. It actually has a mobility that's 100 times higher than silicon. So you can imagine it for kind of next generation processing and things like that. But people couldn't figure out for the longest time exactly how to isolate one of these single layers of graphene from this graphite composite here. And the way that in 2004 they figured out how to do it, which is they invested in some Scotch tape from CVS, and they basically were able to peel off just a single, the top layer of the graphene there, and you can just print it onto a silicon wafer like that. So this won the Nobel Prize in 2010 after they made that discovery in 2004.
Does graphene occur naturally anywhere?
Yeah, I mean, anytime, even when you write, you're basically going to have a few here and there, like a single layers of.
So anywhere there's graphite, there's some graphene.
Yeah, basically, yeah. So, but what happens is in the full graphite structure, the electronics are basically being dampened by the fact that you have poor electronics, poor mechanics in this direction, but incredible mechanics and electronics in that direction. So by isolating that single sheet, that's where the magic happens.
How do you get just a single sheet, not 2 sheets or 3 sheets?
You get the whole thing. But the point is they were able to go under the microscope and using nanoscale tools to be able to see where you had like a single sheet jutting out from 2 and then 3. You can see the color change actually as you go down. And so you can basically just isolate the single sheet part of it and do your measurements there. These days you can grow them over large areas by using a process called CVD. You can actually grow it over an entire wafer. Okay, so they don't use the Scotch tape method anymore.
I pointed me to graphenesupermarket.com the last time we talked.
We buy ours from Graphene Supermarket. So.
All based around
right next to Whole Foods. What?
All based around all of it.
Yeah, it's right next to Whole Foods. Okay, so what's the point? Are we just using graphene because it's a hot material, or is there some reason for it? And this gets back to the original properties we wanted to look at before. So this is a nanoscale material. It's a single atom thick. So what exactly does that give you? And the first thing it gives you is it gives you incredible mechanics, because the bond between the carbon here and any one of these carbons and the substrate is pretty weak. It's called a van der Waals bond. But the thing is, you're integrating this over an entire sheet, and also the sheet is incredibly flexible, so it can conform to any kind of rugged surface here. So they did these kind of bubble tests here to see exactly what the adhesion between the graphene and the substrate is, and it actually has to be. It actually turns out to be the same as a liquid solid adhesion. So it's an incredibly strong adhesion. So when you think about this in terms of a device that you're putting on the body, this is something that will adhere pretty well to the body. And then the second thing is, because every atom is a surface atom, as a sensor, anything that binds to this graphene is going to basically, it's binding to that single, it's binding to the entire structure of the graphene here. So it's really going to modulate the electronic properties of the graphene and give you some sort of response. And that's what we'll see as we go along here. Yeah, question. Okay. So the next question is, what kind of temporary tattoo platform do we use here? And so this is a silk moth here. And it turns out silk moths, there's going to be a lot of animals in this talk. This is just going to be the first of many. And it turns out the silk moth is just like a dog. It's not something that developed naturally over time. It was basically as kind of grew as people trying to produce as much silk as possible from the silk moth to produce clothes in China for the past 5,000 years. But what we can do in our lab is get a bunch of undergrads to buy these silk cocoons here, and basically they'll spend some time in the back of the building cutting these things open and dumping the worm out. And what you can do is you can actually dissolve those cocoons in a solution here and form this solution of silk that you can then process into, it's kind of like a gummy type of solution, and you can basically process it out into thin films, and they kind of look like transparency films, but they're a little bit sticky. And that's kind of the key thing about this silk, which is it's basically soluble in water. So it's just like any kind of temporary tattoo substrate where you want to put your device on this substrate, but then you want the substrate itself to dissolve away. So it turns out silk from a silk moth is actually what we use as this kind of temporary tattoo platform. It's nice and water soluble. I should have brought some today. Actually, I gave a talk once where I wore a few of them on my wrist here. But
any allergen issues with that stuff?
Not so much. It's pretty biocompatible, and it doesn't irritate the skin at all. At least when I wore it for like 24 hours. So, okay. So what you can do is you can take. We talked before. You can print graphene from the original graphite using Scotch tape, and what you can do is if you wet the silk a little bit, it gets a little bit sticky. It's kind of like glue. So you can print graphene on here. It kind of looks like someone scribbled with pencil on the silk here. You can print multiple times these kind of graphene sheets on there. And then you can basically you can dissolve away that silk and then transfer your graphene and maybe some electrodes. Here's 2 gold electrodes and here's a graphene, let's say, onto a tooth. And this is our collaborators tooth here. I have no idea why he had this available. We said we needed a tooth for this project. He's in his 40s, and he said, okay, I'll send you one tomorrow. We got it the next day. I have no idea like why. But anyway, I didn't ask any questions. And then, yeah, we put $1 under his pillow afterwards. And so then also, what was kind of cool, we have a little logo for this now, because this was in last week's Sunday New York Times Magazine. I also didn't know they were going to do this. Someone emailed me and said, hey, you're on the cover of this. I thought that was kind of cool, this kind of smart teeth thing. So so this is just basically a device with 2 electrodes, but this doesn't get around our other issue that we want, which is basically to have remote power and remote readout here as well. So one of the things we've been working on is integrating, this is the device, so you'd have a graphene sheet here, and then you'd have electrodes basically that contact the graphene, and then you have some sort of coil that you can use for the wireless readout. But the problem is that coil is kind of big, and so unfortunately we couldn't use a human tooth for this part, so we had the same undergrads go and get a cow tooth from the local farm. That's one of the nice things about being in kind of farm territory. You can just get some animal parts whenever you need them. So you can interface this thing onto a cow tooth here. You can also interface it anywhere onto an IV bag. Costco cheese was a recent problem with bacterial contamination, so we put it on Costco cheese. And you can also, of course, put it directly on the skin if you'd like. So this is what the device looks like when you interface it onto the skin there. And it seems to hold pretty well if you do it right. And that's, again, to this high adhesion of the graphene, the fact that these are nanoscale materials, so they have very high surface area, so they're going to conform pretty well to any substrate there. So in terms of stability, we took the tooth and we put it in Listerine, which is popular among cows. So we put this tooth in Listerine and we swirled it around for a while to see exactly what happened. If you take the tooth out of the Listerine, surprisingly the device stays on there. And you can go in and you can do a technique called Raman spectroscopy on the graphene there. You can see that the graphene itself is not too damaged. This peak, if you don't know much about Raman, that's fine. It's just this peak goes up a little bit, which means the graphene gets a little bit damaged on the tooth, but it's actually not too much. The device as a whole stays intact, even due to this pretty harsh chemical and mechanical swirling here. Of course, if you try to brush it, it's going to brush off. So it's robust up to a point. Okay, but they're pretty cheap devices, so you can just put another one on after that. Okay, so what the next question is, what do you want to tailor this for? What do you want to use it for? And in fact, this is a platform that can be tailored for anything. In our case, what we've been interested in is looking at bacterial contamination, because this has been in the news a lot recently, because it's a problem in both developing countries where they have poor sanitation, and also in developed countries where basically we consume so much meat and dairy that we're basically have a lot of E. coli contamination in our food. And so you constantly hear stories in the news about person-to-person, food-to-person, bacterial, pathogenic issues. And so this is something that I thought would be an interesting problem to look at. And this is kind of more of the chemistry end of things. We'll get to this sort of more electrical engineering and MEM stuff a little bit later. But in terms of sensitivity, I mentioned before that graphene is this single sheet. So every atom is a surface atom, so anything that binds to it should modulate the conductance. And the thing is, in terms of bacteria, you want the sensitivity to be as high as possible, because it only takes a few bacteria to make you sick, like on the order of 10. Very small number of bacteria that can go in the body, amplify, and make you sick. And so the way that they do this now is you take a sample of water, you have to send it away to a lab, and they do something called PCR on it to get down to those levels of bacteria. But what we want is a real-time device that can sense at very low concentrations, ideally the single cell level. So in this movie here that I have, we're actually focused in on the graphene plane. We're in the plane here, and then we dye our bacteria red. So this bacteria comes along here, and then boom, it just binds to the graphene right there. So we're monitoring this optically. This bacteria comes, single cell bacteria comes, binds to the graphene here, sits there for a while, and then basically gets up and leaves. And if you monitor the conductance of this graphene sheet at the same time that you're watching this movie, you can actually see that when that bacteria binds, the conductance drops, increases actually here, and because this is resistance. So the conductance increases, then when the bacteria comes off, it actually goes back down to the base level. So what's happening here is this is actually just like a transistor, which I assume most of you guys know. It's a 3-terminal device. One of them is called one of the terminals is called a gate, which is like a a faucet on a sink, which basically modulates the conductance in your channel. The same thing is happening here. The bacteria is actually negatively charged. So that it comes down and binds to that graphene, modulates the conductance, and that's basically, it's acting exactly like a field effect transistor here in terms of this device. So it's basically modulating the conductance there. So a single bacteria is enough basically to get a real time. This is less sub-second response here, around maybe 1 second response here that you can actually measure down to the single cell level. So it's pretty impressive device here. Yeah.
Ballpark the conductance that we're talking about for in my digital world. There's conductive and not conductive, which is like copper and air. Like what are we talking? Are we talking about going from really conductive to a little bit less conductive, or in the grand scheme of what we're used to, is this all not superconductive?
Yeah. So the resistance here is on the order of the kilo ohm level. So that's pretty typical for nanoscale devices. In this case, this is just a single cell, so you're talking about a 1% change in that. But the thing is, you can, if you had multiple cells, you'd see a much more significant change, like 10% or things like that. So on that scale, yeah. Mary?
So if there's one bacteria that touches the device, then you can sense it. But I guess I'm wondering if you're eating a burger that's infected with E. coli and there's some bacteria in it, what's the probability that one of those bacteria will come in contact with the small region of the one tooth that has it? Like, are there so many bacteria in the burger that it is, or is it still kind of a low probability event that the bacteria gets to the right?
Well, I guess what you can do is you can put one of these on each one of your teeth to maximize the probability, and I guess it depends on the way you chew too. But yeah, they would have to come in contact with it and basically form enough of a residence time here that you'd basically be able to see this response. So yeah, we haven't done any real-time human studies on this sort of thing, but I'll show you one real-time movie a little bit later, but nothing where we actually put it directly on someone's tooth while he's chewing and things like that. But that would be a next stage. Another thing, yeah.
Processing equipment as well.
What was that?
You could add it to processing equipment, the storage equipment.
Yeah. So a lot of people, they make these sensors that are N by N, very large numbers of sensors. They come with a fingerprint. What we're trying to do here is a little bit different. We want to try to maximize selectivity. That's just interesting for us from the material side of things. But because you don't want to put the burden too much on the processing, you want to have some burden on the sensor itself. So, but this is one of the trade-offs that has to be addressed. Yeah. So, any more questions? Okay. Yeah.
I don't know if this is even allowed, but does this change in conductivity depend on what bacteria infects or touches the surface?
Yeah. So I don't want to get into the gory details, but turns out that gram-negative are the ones that gram-negative. This is called gram-negative. So that's E. coli, H. pylori, which I'll show you later, and strep. Those are gram-negative. Those tend to be the ones that most infect humans, and those tend to be the most negatively charged. The gram-positive ones, things like staph, actually they have significantly less negative charge, so they won't have as large of a response here. But they're not as harmful to humans. So yeah.
So like less than 1%. So if you have 1K, you have only the change of like 10 ohm.
Yeah.
Now that is responding to one bacteria, right? If you have 10, can you say that it's going to be scaling linearly?
Yeah, because
there will be a threshold for the response of the graphene, meaning you're not going to get like 10% of the change. I mean, that's close to 1%. 10 bacteria doesn't really gonna, we don't know, right?
No, we do, because if you've ever looked at something called a transconductance curve of a field effect transistor, we're right on that curve there. So we're right on the modulation part where a very small change in the gate voltage basically produces a huge change in current. So
you don't have that same response curve here corresponding to the graphene?
Well, we do. We always test our graphene like as a standard transistor first using a wafer back gate before we do these type of experiments. Yeah.
That response that you see.
Yeah.
With the same, I mean, you can see the slope basically.
Yeah, we can see it. Yeah. So you can measure the transconductance and all that. I kind of left out some of the gory stuff here a little bit. Okay, so let's see. So I'm going to go through this part a little bit fast because it's more chemistry type of related. But to get kind of, this is also related to Mary's question, which is basically if you want to isolate bacteria from a group of different populations of bacteria, you need to basically impart some selectivity onto it. And one way to do that is to use antibodies, which occur in your body. They're ways of basically they're used by the body to kind of target certain bacteria, certain viruses, and basically let your body know that they've been targeted. Well, the problem with antibodies, putting antibodies on a device, is that they can denature pretty easily. So they limit the lifetime of the device. So what we use are something called peptides. I'm not going to get too much into the details here, but peptides are basically small-scale versions of antibodies. They only have a few amino acids, maybe 10, whereas antibodies could have thousands of amino acids, and they fold intricately. Whereas these peptides don't fold as much, so they're not as selective as antibodies, but they still provide some degree of selectivity to the device. And so the question is, you need to come up with some sort of peptide sequence that's going to bind to the bacteria that you're interested in. And so this was we came up with this idea here when I was watching a special on PBS about this bird called the red knot. And this is a bird. The show was about how this bird is kind of going extinct, this red knot. And basically, they were trying to figure out why this bird is going extinct. And it turns out that the bird basically spends the winter in Argentina, and then it flies 10,000 miles to the Arctic every year to mate. And I'm not sure why it can't mate in Argentina, which seems like a perfectly good place to mate. But the thing is, it's a very long journey, so it needs to stop at some point and refuel along the journey. And so what it does is it stops at the Delaware Bay near me, and this is why this show was on by me. And so what it does is it feeds on these horseshoe crabs here. So the horseshoe crabs are mating in the Delaware Bay, and so they're producing billions of eggs here, and they're not very monogamous, the horseshoe crabs, but they're basically, as they're producing these eggs here, the birds just come in and start eating the eggs, and this is what they use to refuel on the halfway point of their journey there. And so what was happening to the bird was that the bird was actually going extinct because the number of horseshoe crabs was actually decreasing. So they're trying to figure out, well, why is the horseshoe crab decreasing? And it turns out that there's two reasons for it. The first is that the people use the horseshoe crab as bait for capturing fish. The fishing industry there does. But then the second reason, which they talked about, which I didn't know, but I found fascinating, had to do with the fact that they use the horseshoe crabs in the pharmaceutical industry. And this was something I didn't realize, but it was amazing. What they do is they collect the horseshoe crabs in boats, and they mount them up in the lab. And then what they do is they drain the blood from the horseshoe crab. And so you can see these horseshoe crabs are mounted. This is the state of the art in the pharmaceutical industry. They drain the blood. It happens to be blued because they have hemocyanin. It's copper based. So they drain this blood, and then they throw the horseshoe crab back in the water. And of course, some of the horseshoe crabs die because they drain most of their blood during this process. And so you're probably wondering, well, why are they draining the blood from the horseshoe crab? And it turns out that the horseshoe crab lives in these aquatic environments, so it needs a way to defend itself against bacteria because it's basically in this microbial soup, in this aquatic environment surrounded by bacteria. So the blood is incredibly efficient at basically coagulating around bacteria and then killing them. So what the pharmaceutical industry used to do is they used to take their drugs and products and basically inject them into rabbits, but it took too long to wait to see if the rabbit got sick. So what they found is they could take the horseshoe crab blood and drop it on their drug or their device and see if they see it coagulate under the microscope, they'll know instantly if it's contaminated with bacteria. And so I got some inspiration from this because I figured there's got to be a better way than the horseshoe crab for doing this. This is what they use now in the pharmaceutical industry. So we continue the animal theme here, so I read this other article about these African clawed frogs here, which also live in aquatic environments. And they're called clawed frogs because they have claws actually on their hind legs. They don't have teeth, so they use the claws to tear their food apart. So the way that these guys defend, they don't have the kind of blood that horseshoe crabs do that kill the bacteria, but what they do have are these peptides, kind of like the ones I showed before, that are on the skin of the frog. And that's what basically acts as a first line of defense against bacteria. They're called antimicrobial peptides. In fact, humans have them, for example, in our eyes that act as a defense mechanism against bacteria. And the nice thing about it is, unlike, you could not put an antibody, antibodies exist in the body. They're regulated in the blood. That's how they can remain folded. You couldn't put one of those on your skin because they would basically denature. But a peptide you can put on the skin. So that's why the frog has peptides on its skin. and they can basically defend the frog from bacteria. So what we did is we didn't kill any frogs, but we took the sequence that occurs on the frog skin and synthesized that in the lab, and you came up with this peptide sequence here. This is what it looks like. This is the antimicrobial peptide component. These are on this end here that came from the frog sequence, and you can basically put this on your graphene device, and it'll basically just assemble naturally across the sheet of the graphene. So you have your peptides here, your graphene surface underneath, and then the bacteria will come and bind to that, and basically it can act as a target for these bacteria. So this is what enabled us to go beyond the kind of detecting any kind of bacteria that binded like that I showed before, and actually doing some more selective measurements. So we were able to actually find an antimicrobial peptide that binds to H. pylori, which occurs in your saliva, because again, we're talking about teeth here, so we're interested in saliva and breath disease detection. So these occur in your saliva and are indicators of stomach and stomach ulcers and stomach cancers. There's H. pylori bacteria here. So we were able to put the graphene on this tooth here, this cow tooth, and then basically put a matching coil on top of it that matches this tuned to match the coil that's actually on the tooth there. And we can monitor in real time the detection of H. pylori here from a sample of saliva. So this is bare saliva. You can see the saliva on its own with no bacteria doesn't produce a very measurable response in the device. But as you add H. pylori to that saliva, you can see you start to get this much more significant conductance change. Here the concentration is a lot higher, so we're going to like the 10% conductance change level. And so you can answer, to address the question from before, as the H. pylori concentration increases, you get up to a 25% change in the conductance. And we can go down to the 10 to the 2 level. This is wireless. You lose a little bit with the wireless coupling. So we can't do single bacteria resolution, but we can do 100 bacteria, which is actually still below the amount that it takes to get you infected with H. pylori. Yeah. Question?
Yeah. You answered. You just reading the Q of the tank change as the bacteria shows up?
Okay. Yeah. So we collaborate with the electrical engineer on this, and because you're changing the conductance here. Usually, if you have an LC circuit, you want to change either the capacitance or the inductance. But what's happening is the resistance is changing in this device. So what we're actually measuring is we're measuring a change in the bandwidth of the device rather than a shift in the resonant frequency. Yeah. It turns out that also seems to be less susceptible to like bumping and moving of the coil relative to the device. Yeah. This was something that my student worked out with an electrical engineer named Naveen Verma at Princeton. Okay, so this is bacteria, and then the one last thing I want to show you related to this part here, which we're at the halfway point. Anyway, is this is the real-time measurement. So this is not there's no peptides on this device. This is just a bare tooth there. But the thing is, this graphene is extremely sensitive even to water and other volatile molecules here. Oops. So let's try that again. Turn the volume down. So that's the measurement. There's our cow tooth just sitting there freestanding on the table. You can see there's no wires coming out of the cow tooth. This is our baseline conductance. He filmed this with a potato, and there's the coil there. So my student just breathed on the tooth, and you can see the conductance jumps up quite quickly when he breathes on it in this kind of wireless platform here. So that's just water molecules and other molecules from the breath interacting with the tooth there. And then as they desorb off the tooth, it drops back down to the baseline, and it can come back in again, and he can breathe on the tooth a second time, and it'll go back up again to this maximal value here. So you can do this multiple times here with this tooth and get this breath detection. So what we're actually doing now is we have samples of breath from asthma patients and from healthy patients, and we're trying to see if we can distinguish those two patients using this kind of platform here. Okay, so you can see yeah.
As a severe asthmatic, I'm kind of curious, what are you using as the to try to detect between asthma and normal?
We're using antibodies. So yeah, but this gets back to the point you were making before, which is people have used again, like less selective things, and we wanted to use something that's a little more controversial. So we wanted to really up the selectivity there to the antibody level. But people have used like large-scale signal processing.
Oh, sure.
Yeah.
Gases.
Yeah.
Things like that. But
Yeah.
this is
Yeah.
this
Exactly.
is a lot nicer.
More questions? Okay.
Yeah.
Is it possible to resolve the conductance from different sources? Because it seems like the breadth affects the conductance, the micro affects the conductance.
Yeah.
So have you done experiments on that?
Yeah, so the way that we get around that is to use like an array of devices, because these are basically microfabricated, the way you would fabricate any kind of computer chip. So the only thing, the thing that's limiting the size here is that coil. If the coil wasn't there, you can make it arbitrarily small. You can make a coil that loops back on itself that's much smaller, but we couldn't figure out how to do that here. So if you make an array of devices, you can tailor each one with a different peptide or different antibody so that it responds to different bacteria, and then basically come up with the sum of those to give you a net response there. Okay. So this is the other side of my group, which has to do with interfacing energy harvesting devices with the body. And the way that where this project came about is I was reading about pacemakers, and it turns out, of course, pacemakers run off of batteries. And so you have this pacemaker, you have to open someone's chest to install this pacemaker. It runs off a battery. Then after 5 years, the battery dies. And so you have to basically have an entire surgery again to replace this entire unit here. They leave these leads in place here, but they have to replace the entire pacemaker there. So this can lead to infections. It's extremely uncomfortable. It's a surgery that's required, and this happens every 5 years. So we thought maybe there's some way that you can harvest energy from some sort of source in order to prolong the life of this battery. And of course, you couldn't put a solar panel on you'd have to put it on your chest and then wear shirts with holes in it or something like that all the time. So we thought we had another concept that we thought about, which is we read this paper here by this guy named Thad Starner, who's currently working on this Google Glass thing at Google. So he used to be a professor at Georgia Tech, but basically he did this paper in 1996 where he showed that we eat about 10 megajoules of energy. We consume 10 megajoules of energy every day. That's about a quarter of a liter of gasoline in terms of energy. So that has to be dissipated somehow. And it turns out that some of it, a lot of it is dissipated by walking. So that's about 70 watts of power available. But then some of it can be dissipated by breathing as well. So if you could harvest some of this mechanical motion of your lungs or your diaphragm to power a pacemaker, I think that would be pretty significant. And these values here are actually pretty commensurate. You guys would know better than I would, but they're commensurate with the kind of powers that portable electronic devices draw these days, which are kind of at the low watt to milliwatt, and a pacemaker is microwatt level in terms of power. So if you can convert this energy as efficiently as possible, you could actually imagine powering this device at least prolonging the battery life. If you can go from 5 years to 10, that's a significant health issue for the patient. So again, this becomes a materials problem from my perspective, because the question is, what kind of material are we going to interface with the lung tissue in order to harvest this power? And it turns out that there's materials called piezoelectrics, which I assume most people know about, which are basically materials that can convert mechanical energy directly into electrical energy. And at the molecular level, the way that they work is this is a quartz crystal here, which is kind of a naturally occurring piezoelectric, and it has a tetrahedral shape to it. But when you compress it, the bottom part of the tetrahedron basically becomes flattened a little bit. So you have a net dipole in this direction in the individual molecule. And of course, windows are made out of silicon dioxide as well, but all these dipoles are randomly oriented in a window. But in a quartz crystal, they all basically stack up. You have a net polarization across the crystal. So this is where the piezoelectric effect comes from. And it can be used in both directions. It can be used as a sensor, which means converting mechanical into electrical. It could also be used as an actuator, which means basically actuating, making little resonant MEMS devices and things like that. So this is the thing we were interested in, and I can't give a talk without having at least one equation, because I am an engineer. But it's a really simple equation, which is how much energy is stored in one of these piezoelectrics. And it turns out it only depends on three things. The first is the volume of the piezo. The more piezo you have, the more energy that's going to be stored in it. It's basically like a capacitive. This is just a capacitive storage equation here. The second one is the stress that you apply. If you have more stress applied to the piezo, it's going to produce more power. Those are both material independent properties. The one that depends on the material is this piezoelectric charge coupling constant here. So that's material dependent. And so you want that D value to be as high as possible to get as much energy stored in the material as possible. And if you look at the list of materials that are available to you, Here's quartz that we mentioned before. Its D value is pretty small. It turns out that the bone in your body is very, very weakly piezoelectric, but it'd be difficult to put like wires all over your bone and power your iPod or something. There's also polymers called PVDF, which are piezoelectric. But the king of all piezoelectrics is PZT, which stands for lead zirconate titanate, which has a massive D33 value and is basically greater than 80% efficient at converting energy. So if you compare this to like the best solar cell, which can basically do maybe 25%, this is significantly higher than that. But the problem with PZT is it gets back to this problem that I discussed on the first slide. It's exactly like silicon. It's hard. It's a brittle material. It shatters easily, and also the P stands for lead. So it contains lead. So of course people remind me of this every day. But the thing is, in most pacemaker batteries contain like cadmium, which is also toxic too. So if you encapsulate it properly, then you should be able to get around that problem. So these are the but this is an issue that has come up a lot, and so I'll discuss our strategies for how we get around all of these problems. Okay, so let's see here. I want to skip a few things, and okay, let's skip to this one. So let me just, if you want some of the nitty-gritty details about how we process these things, I'll tell you, but we can process large-scale arrays of PZT nanowires. So each of these horizontal lines here, these small horizontal lines, is a little PZT nanoribbon here, which we can process over an entire wafer. So you can do this over a wafer scale. And in fact, if you take this wafer and hold it up to the light, you can actually see a diffraction. This is just a photograph. You can see the diffraction pattern of the sunlight off of those nanoscale wires on this wafer here. Yeah.
It's like a ion deposition or graphic.
Okay, yeah. So you want the nitty-gritty. So basically what we do is we do a process called sputtering. You can also do sol gel. That's actually one of the nice things about PZT is you can produce it in liquid form. So you can actually like put a drop of it on a wafer, spin the wafer, and have like a massive. Oh, okay. Yeah. So you've done. Yeah, exactly. So it's coming. It's come back now because the reason why this is. I kind of skipped over this, but the reason why this is interesting from a material standpoint, the reason why I got into it is because people have made nanowires now of every single possible material, but they usually grow them from the bottom up. PZT, you can't do that because you have the lead needs to be, and the zirconium and titanium all need to be in exactly the right stoichiometry in order to get the maximum piezoelectric response. You just can't grow it like that way. So we start with the film, like you're saying, and then carve that film up into wires like this. Yeah. Okay. So the question is, well, again, why nano? Why do you need nanowires here? And it turns out the answer is that if you take the answer can be found in optical fibers. So of course, a window pane is not flexible because it's kind of a thick material, and you can't bend it very far before it shatters. But if you have the mechanics of any material are such that if you scale it down so that it's extremely thin and extremely long, like an optical fiber, which is also made of glass, you can bend it very easily as if it was just like a pasta noodle or something like that. So this is the motivation from a mechanics standpoint of going to the nanoscale level, which is basically you can increase that mechanical flexibility quite significantly. And also there may be some interesting effects too as you scale it to the nanometer scale, like for example, improvements in piezoelectric response just from this scaling. So that's more of the sciencey side of what we do. But then the question is, again, PZT, the same problem as with silicon. You need to process it at 600 degrees in order to make it piezoelectric. So that's obviously not compatible with the body. So the question is, how do we get, this helps us get around the mechanics by doing this nanoscale patterning, but then how do we get around this temperature problem? And the answer is it's a very, very simple technique. It's exactly like printing newspaper ink onto silly putty, which is we basically have these PZT nanoribbons here, as we call them. They're basically, they're called ribbons because they're kind of like a little bit wider than they are thick. And so they're kind of like ribbony shaped. And they're on this substrate here, this starting substrate. So you can process them at very high temperatures to make it nice and the nice stoichiometry, the highest in terms of piezoelectric response. But then you can take this piece of silicone, this PDMS, it's just like silly putty, which is also silicone, and you can print it down onto these ribbons and literally peel it off. And you'll basically be peeling it off that substrate there, and you can basically print these things directly onto the next substrate here. So again, exactly like printing newspaper ink on a silly putty, that's all it is. Because this process here is done at room temperature. So you can basically process the PZT first at the high temperature and then do the printing at room temperature. And you can see here that before and after, before printing and after printing, that the ribbons actually don't break at all during this process. And they don't get moved around or anything. They go perfectly before and they're perfectly after in terms of this printing. And of course, just like with a newspaper, if you have a large piece of silly putty, you can print the entire newspaper page without any more significant effort. So this was something that we did this for the Time magazine. Actually wanted this somehow. They decided this was one of the inventions of the year in 2010. They must have not been too many interesting inventions that year. But we sent them this chip. So this is actually a large chip that we made, about hand-sized. Here, this is a photograph where you can see these ribbons have been printed over this entire substrate here. So these are all nanoscale ribbons, but it's over the entire substrate, so you can see them all by eye in this photograph. And you can see there's actually some that are missing here. And the reason for that is because they took the chip and they put it face down, and some of them peeled off. They screwed up when they were, we gave them the chip in perfect shape form, but they messed it up here. And then when they sent it back to us, they sent it back face down totally, so they all came off. But anyway, so you can scale this to large areas here is the point. And then it's piezoelectric, so it's flexible and can produce power. I'll show you that to you later. But the question is, I mentioned this before, it's one thing to be flexible, your water bottle is flexible, but even the plastic in your water bottle is not stretchable, right? Your skin is actually stretchable, it can stretch to maybe 20%, okay? So the question is, even though these are nanoscale ribbons, can they be stretched at all, or will they break? And I'll leave it to you. What do you think the answer is? Will they break or not? Who says they break? Okay, when you stretch them. Who says they don't? Okay, so here's the answer. The answer is they do break. So even as you scale them down to nanometer scale, just by stretching them a little bit, they're going to crack up. If you listen closely, it's kind of like sounds like Rice Krispies as they shatter on the surface here. So these ribbons are basically shattering up this direction because we're stretching it in this direction. So we needed a way to kind of get around this problem and make these actually stretchable. And so it turns out there's a classic problem in mechanics where if you take a balloon that's inflated and you put some rigid paint around the surface of the balloon, the circumference here, if you deflate the balloon, what happens is the paint will actually basically buckle up on the balloon surface here and form these kind of corrugated structures here, which depends on how fast you deflate the balloon here. So we saw this. This looks like a fun paper to read, but you can't get past the first page because they go through every possible equation that you can imagine for paint on a balloon. And the interesting thing about this, of course, if you then reinflate the balloon, the paint doesn't break. What happens is it goes from buckled to back to the flat state here. It's this kind of nice state here. If you inflate it from this point on, it will start to crack, but you can go between here and there by inflating and reflating, which is a stretching process, and without any issue. This is something called constrained Euler buckling. If you're interested in this, where Euler buckling is buckling of a beam, and the constraint here is that there's adhesion between the paint and the balloon, which is what leads to this buckling phenomenon. And so we can do the exact same thing here because we print this thing like we do on the silly putty, but our silicone that we use is actually a little bit stretchable. So if you stretch it first and then print it down and then release the ribbons and then release the stretching part of it, what will happen is the ribbons will actually buckle up off of the substrate there. And you can see this, here's a little logo for it, but you can see that this is the flat part of the ribbon and then it'll buckle up off the substrate, and then it goes flat and then buckles up here. And by patterning the substrate there, you can actually control where it's anchored to the substrate and where it kind of buckles off, and there's kind of gaps in between. So you can get these kind of buckled formations here. And now when you stretch it, instead of breaking, you start with these buckled PCT ribbons here. Now you stretch it out. What they'll do is they'll just flatten down onto the substrate here. You can stretch up to maybe 10%, which is almost where skin can go here, and they'll flatten down, and then as you release that stretch, it'll just buckle back up. And you can cycle this multiple times here with the stretch and release type of process. So the cool thing about this is that as you stretch this thing out, you're basically straining the ribbons here. So you're creating a piezoelectric response here as you stretch these things and then release them as well. So you can basically create little energy harvesting generators that work in this kind of stretching mode here, and interface them with the tissue. Let's see how much time do we have. Okay, so but there's one other thing that we've been able to do with these devices, which we've shown recently, and it has to do with the fact that we've actually started to interface cells with them. And this gets back to this question of, are they biocompatible? Can you interface cells with these kind of lead-containing materials? So we found this paper in the literature. We're looking at some of these mechanics of these buckled ribbons, and we found that cells actually have a very similar type of mechanics because they're also curved just like a buckle in a PZT ribbon. And this affects their mechanical properties quite significantly because if you inject current into a cell, let's say this is a neuron. So we all know that brains are electrical tissues. They communicate information electrically, but it turns out that there's some mechanics there too, because if you inject current into a neuron, what happens is you get a polarization difference across the membrane of the neuron, and that leads to a tension in the membrane, which basically forces the cell to swell a little bit. And people have measured this before using something called AFM, which is basically just a sharp nanotip that they puncture into the cell here. And it's exactly like having clothes on a clothesline. If you pull on the clothesline, the clothes will go up, and it's the same thing here. It'll push this tip up. So it turns out that our brains are not just electronics, but also they have moving parts too. These cells will deform a little bit when you electrically spike them. So this was some work that we found in literature where they were able to spike them. This is just this experiment in action. They spiked them using a patch clamp here. They spiked the cell electrically, and they saw that the AFM probe goes up every time they spike the cell. So the cell is responding mechanically to this electrical stimulation, which is an amazing thing. This has not been studied very much in literature. This is the only paper we found, which is about 10 years ago. They saw the cell mechanically deform every time they injected it with current. Yeah.
Like a real mechanical response in the structural response, or is that because fluid is moving in and out and it's puffing up and down?
It's actual mechanical response. There's no fluid moving in and out. Actually, I take that back. There has to be some fluid moving in and out, but it's actually a change in the tension of the membrane. So the membrane has charges in it. You're injecting charge; those interact with the membrane, and then that's what forces it. That tension change forces the volume to change. Yeah.
Nobody followed up on that.
No.
It's pretty amazing.
Yeah. Until we did recently. So this is a paper this is our next thing that's coming out in about 2 weeks. It just got accepted to Nature Nano. So I'm really excited about this stuff. Where we thought, this probe here is pretty invasive. This AFM tip it's a nanoscale tip, and it's pretty invasive. So we thought, well, our stuff is maybe semi-non-invasive. I mean, it does contain lead and everything, but you can imagine culturing a neuron, let's say, onto one of these PZT ribbons, and let's say we suspend the ribbon so there's nothing underneath, so it can easily bend up and down. And now, if you inject current using this pipette into the neuron here, if that neuron expands, it'll push the ribbon down. So it pushes this PZT ribbon down. And if it's pushing that down, you're going to see that piezoelectric response using electrical output. So you have electrical input, electrical output, but you're measuring the mechanics of the cell. Yeah.
So you could, in theory, insert that you could replace the PVT with a genetically altering the neuron to be photosensitive and then imparting charge optically across.
Mechanically photosensitive?
Yeah, basically you hit it with the charge. I'm just making sure I understood this.
Yeah.
You can hit it with charge optically. As long as the charge is imparted with a physiological response that would trigger your sensor. Okay. So individual neuron firing basic bioptical stimulation, that work?
Can you stimulate a neuron optically?
Yeah,
you don't care why it's charged, whether it's charged because you injected electrons or because you put electrically photosensitive things on the surface.
Yeah.
You don't care how charge gets there. The neuron will respond the same way no matter what. The PCT will respond the same way no matter what.
Right, but one advantage of this, though, is one of the questions we had when we first started this project is if you didn't have this is a soft cell, the PZT, even if it's nanoscale, is still a little bit hard. So the cell should just expand upward and not downward. So this patch clamp is also kind of providing like a top constraint, so the cell will push down a little bit as well. In that case, I don't know if that will work, but maybe it would. So, okay, so this is the concept that we came up with, and you can see these beautiful results. My student formed these cantilever ribbons here, and then was able to culture these beautiful neurons with nice dendrites and axons and everything on these ribbons here. You can see the size scale. This is the cell here, and then these ribbons are kind of wide, but they're still only a few hundred nanometers thick, so they can deform pretty easily there. And then each side here we have electrode here and electrode there to measure the piezo response output. Okay, and so first question, of course, PZT contains lead. So neurons like to die. If you've ever tried to culture neurons before, they really like to die. So you can imagine if you put it on a lead surface that they should really, really want to die. But to our surprise, they didn't, which was really surprising. And so we culture these neurons on these PZT ribbons here. The green means that they're still alive. The red means that they're dead. This is after 72 hours. This is after 168 hours. And you can see some of them do die after a longer period of time, but they also die on a standard culture medium, which is the blue here. So compared to PZT, you can grow them on PZT, and they are healthy and alive on this PZT sample. This still doesn't mean you would want to necessarily implant it on the body. But the thing is, everything we've done to this date has these ribbons on a surface. So you can basically put another thin layer of silicone on top to encapsulate it. Basically just spin coat the silicone and then cure it, and then it should be encapsulated. But studies still need to be done to show that that won't leak lead or anything like that. But people have shown that the buckled, even if you have a buckled system and you encapsulate it, that you'll still have the buckling work the way it should. Yeah.
Anyway, you still have some kind of adhesion problem anyway. I mean, we haven't talked about adhesion at all, but presumably there needs to be some kind of coating for adhesion anyway.
Yeah. Okay, that's exactly right. So it's a very weak adhesion between PZT and the silicone, and we know that because we were doing some other studies where we saw we could actually push them around, or just like that Time magazine, if they put it face down, they all come off. So it's a pretty weak adhesion. So you would still want to encapsulate them just strictly for that reason as well, because presumably you're going to be tapping on these things or stressing them in some way. You don't want that to be the weak point, because there's lots of other weak points here too. The cells fire. This is just a standard cell. This shows again that they're healthy, that you can get an action potential in the neuron on the PZT. And so here's the response. So here's our cell that's been cultured on the ribbon. Here's our patch clamp probe that comes down to inject current. Here's our PZT ribbons here. And so we inject the current, and what do we see? So here's the current that's being injected in green here. And amazingly, you can see this perfectly in-phase coherent response from the PZT ribbons to this injected current. You can see it's AC, the way all piezoelectrics basically respond AC because you bend it and then release. And so you get these piezoelectric spikes here every time you spike the cell. So pretty remarkably, we are able to actually measure the mechanics of these cells using this electrical stimulation here. And the other cool thing is you can go in and you can actually quantify this by taking just another one of these AFM tips and pushing down on the ribbon to see how, basically quantify the force versus displacement curve. And so you can translate back from that to see exactly how much displacement, quantitative displacement you're seeing. So here's what happens if you zoom in on the plots here. The green again is injecting the current into the cell, and then the blue is your piezoelectric response. We remove the noise here. You can see this incredibly beautiful response, AC response, that lines up perfectly this lower peak with that upper part, and then the upper peak with the other part of the response there. And if you quantify it, we actually saw that we got a force being acted on from the cell on the PZT of the nanonewton scale, and a deflection of about a nanometer. This actually matches exactly with the results that they got with that AFM tip from that paper 10 years ago. So amazingly, this completely different system here, we're actually able to quantify the exact same response, mechanical response from these cells. So we're really excited about this. But then, of course, we haven't learned anything new from those AFM studies they did before. So the question is, what's new here? And the answer is, you can't scale up this AFM, but you can scale up our ribbons to large areas. So you can scale them up, you can put interdigitated electrodes on them. And we've gotten good now at scaling these things. So here's a wafer, this is a 100 micron scale. The vertical lines here are these ribbons here, and then these are some electrodes that we're putting on them in the horizontal direction there, and on these ribbons. And you can scale this up to a full device here that you can bend and basically generate power. So you can take this device and do what we've been wanting to do from the start, which is basically interface them here with lung tissue. So we got got undergrad to get a cow lung this time, which is significantly harder than getting a cow tooth because you have to kind of pull it out. So here's the cow lung that's breathing here, and here's the device that's interfaced directly onto this cow lung. So you can see it stressing every time the lung is breathing there. So we can measure the piezoelectric charge or power that's being generated from this process here. And so the voltage is actually pretty decent. It's about a volt peak to peak, but the current is still a little bit on the low side. But of course, these are very small strains. So one of the things we'd like to do is eventually scale this up to a level that's enough to generate enough power to power a pacemaker. And I think we can do that because this is a very small device. So we have a lot more area we can go here to generate more power. And so that's kind of one of the things we're interested in doing. And so this is the direct biointerfacing of this device.
You expect kind of linear in size. Power is roughly linear in size.
Yeah, it goes back to the equation from before. This one, V is the volume. So usually you're not going to scale it in the thickness direction because if you do, you're going to lose the flexibility. So the only way you can go is the surface area. Yeah. So so if you do that, then it is linear. It's square with the stress. So this kind of usually PZT crystals are hard. So that's why people haven't put them in shoes, because if you put one of these crystals in your shoe, if you step on it, you're not going to be able to compress them. They basically have the same Young's modulus as steel. So it's like compressing steel with the heel of your shoe. You're not going to do it very much. But the stress here is nice because it's also rod shaped, so you can bend it a lot more stress than you could with a compression mode type of device here. So that's going as a square, and then that surface area is going linear. Yeah. So that's one of the things we like to do. Okay, so I'd like to thank my group and then answer any questions. We're exactly 1 hour. So the people that did some of the work here, Manu did all the tooth stuff, Manu Manor, and he's been a really great person to have in the group. And Yong is the guy who's working on the asthma samples now, which is a really hard project. So he's very brave for doing that. And Fan is the person that did a lot of the piezoelectric ribbon work, especially with the stuff with the cells, with that paper that came out recently. And we've gotten funding from a number of different places. We've worked with companies before, Lockheed, and also with DuPont here, and then also these government agencies as well. And some of our collaborators here, particularly Fio, that's the tooth came from Fio Almanetto. He's a professor at Tufts. And Naveen was the guy who did a lot of the electrical engineering of those wireless circuits from before. So I'd be happy to answer any more questions that you guys have, and thanks a lot. Maybe a question down. Yeah.
Is the phenomenon invertible? Can you induce the charge to get like microfluidics out of this, or?
Yeah. So you can basically, you mean the actuator, like an actuator?
I'm just wondering, since you're responding to the curve, can you impart voltage to get like micro defibrillation of your heart? I mean, I wouldn't use it for that, but like kind of wiggle tissue by wiggle.
Yeah, that's one thing we've been interested in, and I mean, I don't want to give away any secrets here because this is going on a public website, but we are interested in doing the inverse and seeing if we can either something like what you described, or maybe even at the cellular level, seeing what kind of response would happen from the cell from actuating it rather than in this kind of sensing mode here. So that's the next realm of interest for us. So I think it's a really interesting area. So yeah. Okay.
Any other questions? Let's thank Mike one more time.
All right. Thanks a lot.